a WikiVidi Documentaries production. Don't forget to like and subscribe. Enjoy. Neural coding. Neural coding is an neuroscience field concerned with characterizing the hypothetical relationship between a stimulus and the individual or ensemble neuronal responses, and the relationship among the electrical activity of the neurons in the ensemble. Based on the theory that sensory and other information is represented in the brain by networks of neurons, it is thought that neurons can encode both digital and analog information. Overview. Neurons are remarkable among the cells of the body in their ability to propagate signals rapidly over large distances. They do this by generating characteristic electrical pulses called action potentials, voltage spikes that can travel down nerve fibers. Sensory neurons change their activities by firing sequences of action potentials in various temporal patterns, with the presence of external sensory stimuli, such as light, sound, taste, smell and touch. It is known that information about the stimulus is encoded in this pattern of action potentials and transmitted into and around the brain. Although action potentials can vary somewhat in duration, amplitude and shape, they are typically treated as identical stereotyped events in neural coding studies. If the brief duration of an action potential is ignored, an action potential sequence, or spike train, can be characterized simply by a series of all or none point events in time. The lengths of interspike intervals between two successive spikes in a spike train often vary, apparently randomly. The study of neural coding involves measuring and characterizing how stimulus attributes, such as light or sound intensity, or motor actions, such as the direction of an arm movement, are represented by neuron action potentials or spikes. In order to describe and analyze neuronal firing, statistical methods and methods of probability theory and stochastic point processes have been widely applied. With the development of large-scale neural recording and decoding technologies, researchers have begun to crack the neural code and have already provided the first glimpse into the real-time neural code as memory is formed and recalled in the hippocampus, a brain region known to be central for memory formation. Neuroscientists have initiated several large-scale brain decoding projects. Encoding and decoding. The link between stimulus and response can be studied from two opposite points of view. Neural encoding refers to the map from stimulus to response. The main focus is to understand how neurons respond to a wide variety of stimuli, and to construct models that attempt to predict responses to other stimuli. Neural decoding refers to the reverse map, from response to stimulus, and the challenge is to reconstruct a stimulus, or certain aspects of that stimulus, from the spike sequences it evokes. Hypothesized coding schemes. A sequence, or train, of spikes may contain information based on different coding schemes. In motor neurons, for example, the strength at which an innervated muscle is contracted depends solely on the firing rate, the average number of spikes per unit time. At the other end, a complex temporal code is based on the precise timing of single spikes. They may be locked to an external stimulus such as in the visual and auditory system, or be generated intrinsically by the neural circuitry. Whether neurons use rate coding or temporal coding is a topic of intense debate within the neuroscience community, even though there is no clear definition of what these terms mean. In one theory, termed neuroelectrodynamics, the following coding schemes are all considered to be epiphenomena, replaced instead by molecular changes reflecting the spatial distribution of electric fields within neurons as a result of the broad electromagnetic spectrum of action potentials, and manifested in information as spike directivity. Rate coding. The rate coding model of neuronal firing communication states that as the intensity of a stimulus increases, the frequency or rate of action potentials, or spike firing, increases. Rate coding is sometimes called frequency coding. Rate coding is a traditional coding scheme, assuming that most, if not all, information about the stimulus is contained in the firing rate of the neuron. Because the sequence of action potentials generated by a given stimulus varies from trial to trial, neuronal responses are typically treated statistically or probabilistically. They may be characterized by firing rates, rather than as specific spike sequences. In most sensory systems, the firing rate increases, generally non-linearly, with increasing stimulus intensity. Any information possibly encoded in the temporal structure of the spike train is ignored. Consequently, rate coding is inefficient, but highly robust with respect to the icy noise. During rate coding, precisely calculating firing rate is very important. In fact, the term firing rate has a few different definitions, which refer to different averaging procedures, such as an average over time, or an average over several repetitions of experiment. In rate coding, learning is based on activity dependence and haptic weight modifications. Rate coding was originally shown by Ed Adrian and Wise Ottoman in 1926. In this simple experiment, different weights were hung from a muscle. As the weight of the stimulus increased, the number of spikes recorded from sensory nerves innervating the muscle also increased. From these original experiments, Adrian and Zotterman concluded that action potentials were unitary events, and that the frequency of events, and not individual event magnitude, was the basis for most interneuronal communication. In the following decades, measurement of firing rates became a standard tool for describing the properties of all types of sensory or cortical neurons, partly due to the relative ease of measuring rates experimentally. However, this approach neglects all the information possibly contained in the exact timing of the spikes. During recent years, more and more experimental evidence has suggested that a straightforward firing rate concept based on temporal averaging may be too simplistic to describe brain activity. Spike count rate. The spike count rate, also referred to as temporal average, is obtained by counting the number of spikes that appear during a trial and dividing by the duration of trial. The length t of the time window is set by the experimenter and depends on the type of neuron recorded from enter the stimulus. In practice, to get sensible averages, several spikes should occur within the time window. Typical values are t100 ms or t500 ms, but the duration may also be longer or shorter. The spike count rate can be determined from a single trial, but at the expense of losing all temporal resolution about variations in neural response during the course of the trial. Temporal averaging can work well in cases where the stimulus is constant or slowly varying, and does not require a fast reaction of the organism, and this is the situation usually encountered in experimental protocols. Real-world input, however, is hardly stationary, but often changing on a fast time scale. For example, even when viewing a static image, humans perform saccades, rapid changes of the direction of gaze. The image projected onto the retinal photoreceptors changes therefore every few hundred milliseconds. Despite its shortcomings, the concept of a spike count rate code is widely used not only in experiments, but also in models of neural networks. It has led to the idea that a neuron transforms information about a single input variable into a single continuous output variable. There is a growing body of evidence that in Purkinje neurons, at least, information is not simply encoded in firing, but also in the timing and duration of non-firing, quiescent periods. Time-dependent firing rate. The time-dependent firing rate is defined as the average number of spikes appearing during a short interval between times t and t delta t, divided by the duration of the interval. It works for stationary as well as for time-dependent stimuli. To experimentally measure the time-dependent firing rate, the experimenter records from a neuron while stimulating with some input sequence. The same stimulation sequence is repeated several times and the neuronal response is reported in a peri-stimulus time histogram. The time t is measured with respect to the start of the stimulation sequence. The delta t must be large enough so there are sufficient number of spikes within the interval to obtain a reliable estimate of the average. The number of occurrences of spikes nk summed over all repetitions of the experiment divided by the number k of repetitions is a measure of the typical activity of the neuron between time t and t delta t. A further division by the interval length delta t yields time-dependent firing rate R of the neuron, which is equivalent to the spike density of PSTH. For sufficiently small delta t, R delta t is the average number of spikes occurring between times t and t delta t over multiple trials. If delta t is small, There will never be more than one spike within the interval between t and t delta t on any given trial. This means that r delta t is also the fraction of trials on which a spike occurred between those times. Equivalently, r delta t is the probability that a spike occurs during this time interval. As an experimental procedure, the time-dependent firing rate measure is a useful method to evaluate neuronal activity, in particular in the case of time-dependent stimuli. The obvious problem with this approach is that it cannot be the coding scheme used by neurons in the brain. Neurons cannot wait for the stimuli to repeatedly present in an exactly same manner before generating response. Nevertheless, the experimental time-dependent firing rate measure can make sense if there are large populations of independent neurons that receive the same stimulus. Instead of recording from a population of n neurons in a single run, it is experimentally easier to record from a single neuron and average over n repeated runs. Thus, the time-dependent firing rate coding relies on the implicit assumption that there are always populations of neurons. Temporal coding. When precise spike timing or high-frequency firing rate fluctuations are found to carry information, the neural code is often identified as a temporal code. A number of studies have found that the temporal resolution of the neural code is on a millisecond time scale, indicating that precise spike timing is a significant element in neural coding. Such codes that communicate via the time between spikes are referred to as interpulse interval codes, and have been supported by recent studies. Neurons exhibit high-frequency fluctuations of firing rates which could be noise, or could carry information. Rate coding models suggest that these irregularities are noise, while temporal coding models suggest that they encode information. If the nervous system only used rate codes to convey information, a more consistent, regular firing rate would have been evolutionarily advantageous, and neurons would have utilized this code over other less robust options. Temporal coding supplies an alternate explanation for the noise, suggesting that it actually encodes information and affects neural processing. To model this idea, binary symbols can be used to mark the spikes, 1 for a spike, 0 for no spike. Temporal coding allows the sequence 00011000111 to mean something different from 00110011011, even though the mean firing rate is the same for both sequences. At 6 spikes/10 ms until recently, scientists had put the most emphasis on rate encoding as an explanation for postsynaptic potential patterns. However, functions of the brain are more temporally precise than the use of only rate encoding seems to allow. In other words, essential information could be lost due to the inability of the rate code to capture all the available information of the spike train. In addition, responses are different enough between similar stimuli to suggest that the distinct patterns of spikes contain a higher volume of information than is possible to include in a rate code. Temporal codes employ those features of the spiking activity that cannot be described by the firing rate. For example, time to first spike after the stimulus onset, characteristics based on the second and higher statistical moments of the IC probability distribution, spike randomness, or precisely timed groups of spikes are candidates for temporal codes, as there is no absolute time reference in the nervous system. The information is carried either in terms of the relative timing of spikes in a population of neurons or with respect to an ongoing brain oscillation. One way in which temporal codes are decoded, in presence of neural oscillations, is that spikes occurring at specific phases of an oscillatory cycle are more effective in depolarizing the postsynaptic neuron. The temporal structure of a spike train, or firing rate evoked by a stimulus, is determined both by the dynamics of the stimulus and by the nature of the neural encoding process. Stimuli that change rapidly tend to generate precisely timed spikes, and rapidly changing firing rates no matter what neural coding strategy is being used. Temporal coding refers to temporal precision in the response that does not arise solely from the dynamics of the stimulus, but that nevertheless relates to properties of the stimulus. The interplay between stimulus and encoding dynamics makes the identification of a temporal code difficult. In temporal coding, learning can be explained by activity-dependent synaptic delay modifications. The modifications can themselves depend not only on spike rates, but also on spike timing patterns, i.e., can be a special case of spike timing-dependent plasticity. The issue of temporal coding is distinct and independent from the issue of independent spike coding. If each spike is independent of all the other spikes in the train, the temporal character of the neural code is determined by the behavior of time-dependent firing rate R. If R varies slowly with time, the code is typically called a rate code, and if it varies rapidly, the code is called temporal. Temporal coding in sensory systems. For very brief stimuli, a neuron's maximum firing rate may not be fast enough to produce more than a single spike. Due to the density of information about the abbreviated stimulus contained in this single spike, it would seem that the timing of the spike itself would have to convey more information than simply the average frequency of action potentials over a given period of time. This model is especially important for sound localization, which occurs within the brain on the order of milliseconds. The brain must obtain a large quantity of information based on a relatively short neural response. Additionally, if low firing rates on the order of 10 spikes per second must be distinguished from arbitrarily close rate coding for different stimuli, then a neuron trying to discriminate these two stimuli may need to wait for a second or more to accumulate enough information. This is not consistent with numerous organisms which are able to discriminate between stimuli in the time frame of milliseconds, suggesting that a rate code is not the only model at work to account for the fast encoding of visual stimuli. It has been suggested that neurons of the retina encode visual information in the latency time between stimulus onset and first action potential, also called latency to first spike. This type of temporal coding has been shown also in the auditory and somatosensory system. The main drawback of such a coding scheme is its sensitivity to intrinsic neuronal fluctuations in the primary visual cortex of macaques. The timing of the first spike relative to the start of the stimulus was found to provide more information than the interval between spikes. However, the interspike interval could be used to encode additional information, which is especially important when the spike rate reaches its limit, as in high contrast situations. For this reason, temporal coding may play a part in coding defined edges rather than gradual transitions. The mammalian gustatory system is useful for studying temporal coding because of its fairly distinct stimuli and the easily discernible responses of the organism. Temporally encoded information may help an organism discriminate between different tastants of the same category that elicit very similar responses in terms of spike count. The temporal component of the pattern elicited by each tastant may be used to determine its identity. In this way, both rate coding and temporal coding may be used in the gustatory system: rate for basic tastant type, temporal for more specific differentiation. Research on mammalian gustatory system has shown that there is an abundance of information present in temporal patterns across populations of neurons. And this information is different from that which is determined by rate coding schemes. Groups of neurons may synchronize in response to a stimulus, in studies dealing with the front cortical portion of the brain in primates. Precise patterns with short time scales, only a few milliseconds in length, were found across small populations of neurons which correlated with certain information processing behaviors. However, little information could be determined from the patterns. One possible theory is they represented the higher order processing taking place in the brain. As with the visual system, in mitral/tufted cells in the olfactory bulb of mice, first spike latency relative to the start of a sniffing action seemed to encode much of the information about an odor. This strategy of using spike latency allows for rapid identification of a reaction to an odorant. In addition, some mitral/tufted cells have specific firing patterns for given odorants. This type of extra information could help in recognizing a certain odor, but is not completely necessary, as average spike count over the course of the animal's sniffing was also a good identifier. Along the same lines, experiments done with the olfactory system of rabbits showed distinct patterns which correlated with different subsets of odorants, and a similar result was obtained in experiments with the locust olfactory system. Brought to you by WikiVidi Documentaries. Would you like to know more?